When I learned about cell division for the first time, I've always wondered, how does a cell know when to divide? Not only that, but I had even more questions when I learned that they also divide pretty much after a set period of time. It's such an important aspect of life as we know it, but it was just glossed over, a black box if you will. So, what's the clockwork behind this oh so very important component of life? You know what else happens regularly? Your heart beats regularly. Your body somehow knows when to wake up and sleep regularly too. And even in nature, predator-prey populations rise and fall in lockstep with one another. So, oscillations, things that happen regularly over time, aren't just one-off events. They're ubiquitous in biology. Not only that, studying them for curiosity's sake isn't pointless at all. To study the oscillation controls of a cell is to set yourself up to understand why each of the components are necessary. Removing one of the components can lead to cell division diseases such as cancer. Understanding the circadian rhythm can perhaps lead us to understanding and having better sleep, and if we understand how predator and prey populations evolve, we can find better ways to predict and preserve our ecosystems. So in this video, we're going to be building towards the true essence behind these oscillations. And to do that, we're going to build a tool that can take in, as an input, the connections and interactions within a biological network and spit out how the system will behave. Does your biological circuit just delay towards some steady value? Does it spiral into a certain doom? Or does it perpetually rotate around a certain point? Those skeptical among you right now might be questioning as to why we would need such an overcomplicated tool, as we can simply just predict the dynamics of a system using our intuition and logical reasoning. Those more programming savvy amongst you right now might think of a piece of code or an algorithm that does the simulation of the system for us instead. And those of you who are more mathematically inclined might think of a way to cut down all of that computational work into a few simple steps that can reveal so much about the system. So throughout the video, we'll be going through these 3 different approaches together to uncover the dynamics of how frog egg cells divide. And as a bonus gift, what we will discover today also applies to other oscillations in biology. So without further ado, let's get to the setup of this problem. Cells need a sort of indicator to tell it when to divide. This is very similar to how clock signals in electronics set the pace at which things happen. There are 2 main phases of cell division: the actual cell division, mitosis, and the setup leading towards cell division, interphase. In most cells, this phase is divided into smaller phases that check for things like DNA damage, cell size, and replication of DNA. In frog egg cells, those 3 happen all at once in one phase. This is why I selected frog egg cells in particular, since it's the simplest variation of this problem. These 2 phases are activated by 2 different protein signals. A protein called CDK tells the cell to go into mitosis mode, and APC tells the cell to exit mitosis and enter back into interphase. APC and CDK also form relationships with one another. CDK activates APC through a bunch of steps, and APC deactivates CDK. And we'll see soon, this tangle between the 2 is the heart of the cell cycle's oscillation. CDK is a very important signal since it promotes cell division, so it is very heavily regulated by other components. CDK bumbles around the cell without doing anything until it is activated by cyclin. CDC25 tells the CDK to accelerate into cell division, and WEE1 puts the brakes on that. And this balance between braking and accelerating is controlled by the cellular and external environment. In addition, CDK also positively feedbacks to both of these. These little details will be important later. I will also say that this video requires the essence of linear algebra by 3 Blue 1 Brown as a prerequisite. If I were to teach linear algebra from scratch here, it will take forever. Now that we've gone through introducing all the performers in this ballad, let's get to figuring out how they bounce off of one another to create the dance that is cell division. Let's start by looking at this problem as intuitively as possible. Like I said, the heart of the oscillator is the coupling between CDK and APC, so let's examine this centerpiece more closely. In the beginning, both of these are off. Since APC isn't actively trying to turn off CDK, CDK naturally turns on. Once it is on, CDK switches on APC. But once APC is on, it goes ahead and turns off the CDK. Without CDK though, APC can't sustain itself and turns off. Repeating this cycle over and over again. Let's look at that again, but this time we'll write everything out more clearly. Both of them are initially off. CDK turns on, APC turns on, CDK turns off, then APC turns off, and it repeats. Voila! That's an oscillation. We can expand on this model of oscillations by adding another piece from the hidden steps from CDK to APC. This generates 8 possible states. And the curious thing about this is that if you start on a state, all on or all off, you get attracted into the loop. This behavior is known as a limit cycle. It is a trajectory that ensnares the others into converging onto it. But you might be asking yourself, is this even necessary for the cell? It can easily accomplish the same thing by cutting out APC entirely, and just have CDK just directly repress itself. Once CDK turns on, it just turns itself off in a loop. Why would a cell choose to utilize such a more complicated path? All of this, though, is nice and intuitive and simple, but there is one little problem though. None of these models I propose actually capture how cells actually oscillate. Why? Pause the video and try to answer this. Here's a guiding question. What key assumptions did we make about these 3 that are a little too far from reality than we'd like? Be sure to flex in the comments if you got it before I reveal the answer. Well, here's the answer. We assumed on and off states are discretely on and off. Why is that a problem? Well, in cells, there are many of these proteins bouncing around everywhere. They can't just be all on or all off. There are no discrete on or off levels here, only continuous ambiguities. So, in order to explain the real how behind cell division, we must use a tool that can deal with continuous changes, and that is calculus. More specifically though, differential equations. So let's use differential equations to explain why each of these 3 setups behave as they do. Surprisingly, setting them up uses the same brain muscle as the intuitive approach. The central question to setting up differential equations is to ask, how does something change with respect to another? More specifically to our case, how do CDK activation levels change over time? The equal sign in the equation indicates what the answer to this question is. And for our question, the answer to this is the activation due to cyclin and the deactivation through APC. We'll look at the self-repressing model first, since it only involves one type of protein. Then we'll take the intuition from this and apply it to the more complex networks. In the simplest self-repressing model, we can write these 2 as follows. The rate of activation is alpha, and the rate of deactivation is dependent on how much CDK and APC there are. High CDK means more CDK will be available to be deactivated, and high APC means there's more deactivators. We've made a key assumption in this model earlier though. APC is rapidly activated by CDK, so we can just say that the amount of APC that floats around is just a hill function of CDK. Refer back to the previous videos as to why this is. As you can see, by rewriting everything in math, you are forced to make your assumptions explicit, and for good reason too. It is much easier to trace back to an incorrect assumption than to be lost in a sea of blurry intuitions. And this is why I feel strongly that math is a tool that brings intuition into reality. And dang, does that sound cool. Okay, back on topic. This is going to be a review of solving differential equations using Euler's method. I'm trying to make this as biologist-friendly as possible, but it is a nice refresher for those who are very math and CS oriented amongst you. The differential equation is now ready to be plugged in. I'll also rename this function as f, or rate. I'll use these names interchangeably. It is just a function that returns the rate at some level of activation of CDK. Put in a language that computers can understand, if you start at some initial activation level of CDK, the change in concentration after some time step, dt, is equal to the rate times that little time step. This is actually very reminiscent of how we can calculate distance based on velocity. Let's say we move at speed v for some time t, we will move by v times t, distance. Once we move there, we can recalculate the velocity and move to the next points down the line. However, in this analogy, where you are also determines your velocity as well, meaning that just using v times t isn't an accurate description of how we move, since the positions you've just passed have different velocities associated with them. As you can see earlier, the movement is in bursts of speed, which is much more discrete and dynamic than we'd actually like. In order to deal with this problem, the idea is to touch as many positions as possible during our calculations to capture all the velocities, and we can do that by incrementing the time in little steps instead. And this is why earlier I said that dc star is equal to rate times dt. It's to prevent this problem of overshooting. After you've moved in a tiny step, dc star, you take this as your new starting point and you calculate your rate again, based on where you are right now. Here it is again. If you take a look at our equation again, the rate is dependent on what c star you are at. So you can, in fact, just pre-calculate what the rate is going to be at each location. That's the graph you see. It's the same type of graph we saw earlier for the velocity example. We can also plot the arrows here on the diagonal just like the velocity case, but we'll do it for every possible point. Combining these two viewpoints together, you can see there's a region where the rate is positive, increasing. So if you start or land in that region, you'll follow the arrow where you land on and be moved to the right. The reverse is true for the negative regions. You can see you move to the left. And the middle of that is where the rate crosses the x-axis, and it's where things stop moving. This is because a rate of 0 means nothing changes. This point is what we call a fixed point. It is going to play a huge role later. This is because a fixed point determines the long-term behavior of a system. If the fixed point attracts, then the system converges to a steady state. If it repels, then it diverges. If there are multiple fixed points, then the system tends towards an attractive fixed point if it can, and runs away from the repulsive ones. So in effect, it is as if we can just input our initial C star, and then see how it flows along the arrows, as if they were pebbles in a river. The problem with this one protein case, as you can see, is that it doesn't even oscillate, and it just decays towards a point. Now that we have a one protein case now, let's apply our intuition that we've got here to a two protein case. CDK activating APC, and the model is as follows. We add an extra line that asks the question of how APC levels, activation levels, change over time, and that depends on the activation of it due to CDK and APC's own decay. Once again, CDK is generated at the rate of alpha. If there are more APC being active, then more of it will be available for decay, so it's just beta 2 times the concentration of activated APC. Now, APC activation levels depends on two things: how much deactivated APC there are, and the rate at which C switches on A. And like before, the latter is just the Hill function. One thing we can say about the deactivate APC is that if you combine that with the total amount of activated APC, for any time that this system happens at, the total amount is going to be the same since the on turns to offs and the offs turns to ons. For convenience, we'll just name this amount 1 or 100%. And like last time, the deactivation through APC will be modeled pretty similarly. We can summarize these two equations into one like this. This column of stuff is known as a vector, and yup, it's the same vector arrow from physics. What this physically means is that if you plot C star with A star together, the arrows that will fill that space has a horizontal component that tells you how much you're being changed in the C levels, and your vertical will tell you the A levels. And we can play the same game of letting initial conditions flow along a river vectors and see how the system evolves over time. One thing though is that if we set the equation of dc by dt is equal to 0, or in this case f of a and c is 0, the curve that pops out from that relationship tells you where c stops changing. Same idea applies to a. These curves are known as nullines, and where they intersect is where the fixed point is going to be, since both rates are 0 at this point. As you can see, this system also has a fixed point, in which the system now spirals towards. Hey, at least we have some oscillations now. We can also do the same thing for the 3-protein case, and this time it generates sustained oscillations, but it doesn't really match the qualitative pattern of the data. Even though this is off, this is actually a very good example of what is known as the delay oscillator. And as you can see, CDK levels rise, followed by PLK1, which is followed by APC, which represses the CDK and the cycle repeats. This delay between APC and CDK activation is what causes all three of them to oscillate. And I'll let you in on something. We've seen this type of oscillator before in the first video. That's a human-made repressolator. Try to figure out how it works for yourself, and you'll see that these are also delay oscillators. And another important type of delay oscillator in biology is the circadian rhythm for flies. If you take a look at this circuit here, there's a significant delay between the transcription and translation that causes the oscillation delay. So that's great, at least we have working simulations, but as you can see, if we were to hypothetically add more proteins, then you can't exactly plot it anymore since it will be in the 4th dimension. As a consequence, you also can't visualize what happens at the fixed points anymore, hence you lose the ability to predict long-term behavior of the system, which is kinda what we're after. What I'm going to introduce to you now is a mathematical tool that can pretty much solve this problem by just looking at the equations of the rates alone. And what's even cooler is that the basis of this tool comes from a very simple fact in calculus. The more you zoom into a curve, the more it looks like a line. And this is one of the reasons why this tool is called linear stability analysis. So let's start building our intuition in 1D first, of course. Since we're going to be looking at one fixed point at a time, let's just shift the origin towards the fixed point. The axis will be now in terms of x, which is just the distance of c away from the fixed point. This is especially important in higher dimensions since it's going to be really hard wrapping your head around some sort of like 2D, 3D distance away from the origin. If you zoom into the graph at the fixed point, you can pretty much approximate the curve at that point as aligned with a slope of lambda. The equation for this line is simply lambda times x. You can see why this is convenient, since it's not the clunky, full-blown linear approximation, as you can see in this unshaded line here. And lambda is just equal to the derivative of the curve evaluated at that fixed point. This lambda is also known as the eigenvalue of the system. It tells you how the system evolves near the fixed point. The sign tells you if your fixed point is repulsive or attractive, and the magnitude tells you how strong that is. A positive eigenvalue means that the fixed point is repulsive, since the arrows all point away from the fixed point. A negative lambda, on the other hand, says the system will tend towards the fixed point. Notice the negative lambda does not mean that the fixed point will be flipped around and scaled to negative x. It means that the change will be flipped around and scaled negatively. Thus, it scales down the value overall, so it tends towards the fixed point instead of flipping around it. Trying this on our C curve, you can see that lambda is negative, so the system just slowly scales down towards that fixed point. So the whole rate of change equation is much simpler now. dx by dt is not just f of x now, it's just dx by dt equals lambda x. Those more math savvy amongst you can see that x of t is just approximately an exponential explosion or a decay. In fact, I'll boldly claim that if the eigenvalues are real numbers, the oscillations are impossible to happen. Now that we have a more solid grasp, let's apply this to the 2D case. We'll do the same thing and view the whole thing as x again, and this time, c just becomes x1 and a just becomes x2, and the vector distance now just becomes x. So dx vector by dt is equal to df by dx vector, evaluated at that fixed point. One more problem with this though. How do we evaluate a derivative with respect to a vector? Well, let's split the equation up and see what's under the hood. dx1 by dt is just f of x1, x2. The function of f takes in x1 and x2 as an input. To approximate it, you can fix x2 in place and then find the slope as x1 varies. Same idea for x2. This generates an inclined plane that is tangent to the 3D function, and the equation for this tangent plane is fx1, x1, plus fx2, x2, which gets better when you're not far from the fixed point. Now, we can do the same thing to x2, and we have a system of equations. We can factor out both x1 and x2 as a vector, and we have this matrix which describes our rate of change near that fixed point. This is analogous to the 1D case. This matrix is what is known as the Jacobian. For those of you who have watched the essence of linear algebra before, you know that a matrix can scale, flip, rotate, or do something in between those things to a vector. So what the Jacobian does is that for some input x near the fixed point, it calculates the rate of change at that point, which is scaled again by dt and results in the point moving by dx. What the Jacobian did for the rate vector is known as a linear transformation. The columns of the matrix tell you where the unit vectors land, but for a lot of matrices though, you can describe this transformation in another way. If you look closely, some vectors do not get knocked off their span once the transformation happens. They only get scaled up or down. These vectors are known as eigenvectors, and the scaling value is, you guessed it, eigenvalues. It is basically how the system changes along the eigenvector. Much like the 1D line in the first example, a positive eigenvalue repels things away from the fixed point along a 1D line, and a negative eigenvalue attracts things along a 1D line. And since everything is linear here, the change for any other vector not on the 1D axis will just be a combination of how far along we are on each eigenvector. In addition to positive and negative eigenvalues, there can also be imaginary eigenvalues, which corresponds to a rotation, along with the caveat of an imaginary eigenvector. If you have a complex number as an eigenvalue, the real part tells you how much you scale it, and the imaginary part tells you how much it rotates. So as you can see, if you solve the eigenvalue of the Jacobian of CDK APC case, you see that you get a negative real part and an imaginary part, which tells you that this whole thing will spiral into a certain doom. Isn't that really amazing? Now, we've at least confirmed that the oscillation does happen. It's just not sustained, and if it is, it's qualitatively off. So this is where we listen to the wet lab people and look at the results of some experiments. You can see that if you take the circuit and get rid of the CDC25, the circuit doesn't oscillate anymore and just decays to zero, much like the second case. This means that for our model to be accurate, we must incorporate the interactions with CDC25. The way CDC25 turns on CDK is by coming in and doing the biological equivalent of flipping on a switch. This event happens very quickly, so we'll assume that the feedback is very quick and we can just cut the middleman out. If we look at the phase plot of the CDK positive feedback alone without changing APC, you can see that it has 3 fixed points, 2 attractive ones and 1 repulsive one. This is the same as positive autoregulation for those of you who are from the last video. So at a glance, this oscillation works by using the tangle between APC and CDK as the heart of the oscillator, and the CDC25 acts as an anchor that sucks the oscillation into either extreme state, keeping the oscillation alive. You can see that this is true by looking at the phase plot of the system. I'm going to draw only one of the null lines this time, and with this one corresponding to keeping APC activation levels the same. If we freeze APC levels like earlier, let's say right in the middle of this null line, there's going to be 2 fixed points that are attractive, being on either side, meaning that you'll be attracted to either fixed point. In this case, let's just pick the right one. Let's say time goes on and you end up above the range where the line intersects the null line. Suddenly, the whole thing is reversed, and there's only one attractive fixed point on the other side, so your orbit goes towards that. APC levels go down again, and the whole thing just swings again towards the other side. This is what we call a relaxation oscillator, and this is another way to generate sustained oscillations in biology. This type of oscillation is characterized by sharp impulse waves, which we see in the cell cycle result. This, by the way, is how we can model heart pacemaker cells. Links for this and any other paper I cited down in the description. If we look at it in another way, evaluating the eigenvalue at the fixed point gives us a divergent oscillation. However, it can't diverge too far since there are limited numbers of molecules and atoms within the cell, so at some point the trajectory is gonna be pushed back towards the center. Since the trajectory is oscillating out but being pushed back in, this means there is a certain type of orbit that it oscillates but it doesn't diverge nor converge. This is why we have a limit cycle oscillation for this type of system. This is known as the Poincaré-Bendixson theorem, and as a math person, seeing this pop up out of nowhere in biology really amazes me till this day. Last but not least, I'd like to brush on the role of V1. If we take a look at the rate plot of CDK and CDC25, it is a line intersecting 3 points on the curve. However, if something were to go awry and the line no longer intersects these 3 points, there's going to be a compromise into the sustained oscillations. However, with V1, the curve now looks like this, and it's much more robust to changes. So there you have it. That's the secrets to how oscillations happen in life sciences. We now have a way to see how systems in biology behave long term based on just the connections from within their network. Not only that, but we see why it's extremely useful for us to bring math into analyzing biology. It turns the wobbly biological intuitions into concrete, dynamical descriptions of biology. And that's it for this video. Next time is going to focus on the math behind how life can tolerate more than you think. Stay tuned and don't forget to like and subscribe. Prompt out.